morning, Xiangwei.
Good evening, Jennifer. Hi, everybody. Hi, I'm Zhongwei here. I'm one of the clinician scientists in obstetrics and gynecology and IVF specialist. And today, we are very happy to welcome you to a new episode of Making Reproductive Longevity a Reality.
I'm Jennifer Garrison. I'm a faculty member at the Buck Institute and the faculty director for the Global Consortium for Reproductive Longevity and Equality. And the consortium is really designed to build the ecosystem around female reproductive health and equality. And we're doing that by funding both grants and fostering collaboration between scientists and clinicians, industry partners, advocates, funders, basically anyone who's interested in this space. And this webinar series is designed to highlight some of the research that we funded. So today we're going to have two talks from GCRLE grantees on a really exciting topic related to female reproductive aging, namely cell division in egg cells and how errors in that process early in development might actually contribute to egg quality and also reproductive aging later in life. So what we're talking about here is called meiosis, which is the cell division process that eggs go through. And it starts actually when a woman is developing in her mother's womb. And it takes years to mature an egg cell. And so if you think about that for a minute, what that means is that if a woman is pregnant with a female baby, somewhere around 26 weeks of gestation, all of the oocytes that could become her future grandchildren are actually inside of her womb, which sounds like science fiction to some people. And it turns out that errors in the very early stages of cell division or meiosis are what contribute to aneuploidy or changes in chromosome number. And it's not clear whether that's because maternal age is contributing to some, accumulated damage that's happening over time, or whether there's something that's really pre-existing, some difference in the eggs that's pre-existing from the time that they were first made in the womb. So Xiangwei, I know that you screen IVF embryos for chromosome abnormalities before you transfer them. So what kind of trends have you seen in your clinical practice?
Right. So Jennifer, this thing about screening chromosomes in IVF embryos, actually in Singapore, it's still under a restricted research protocol, but throughout the rest of the world, it has been practiced, especially in Europe and the US, some parts of the world. And the lot of cumulative data that I actually have been looking through when I was doing my doctorate as well, and also a lot of recent published data that have examined IVF embryos from women of all ages and demonstrated that even in women less than 25 years old, up to 45 of their blastocysts are aneuploid, which means they carry abnormal numbers of chromosomes. So that's actually quite high. And once a woman hits above 40 years of age, this blastocysts, which are actually day 5 embryos, more than 70% of them are actually aneuploid. So this is very strongly associated with the age of the women. And as you've mentioned so nicely and gives the right context, is that it's probably due to these eggs getting older with a woman as her life progresses. And somehow these eggs don't seem to be allowed, themselves to undergo normal division, as you mentioned, and result in this abnormality finally after the embryo is fertilized. And I think with that, today's two speakers will be given an excellent insight into how this phenomenon works. And I now do want to remind all the audience here to put in your questions in our Q&A segment. And with that, I think let's now proceed to the first speaker.
It's my pleasure to introduce Iain Cheeseman. He's a member of the Whitehead Institute and a professor of biology at MIT. He received his PhD from the University of California at Berkeley, working with David Drubin and Georgiana Barnes. He carried out his postdoctoral research in the lab of Arshad Desai at the Ludwig Institute for Cancer Research in San Diego. And his lab now investigates how core cell division processes are rewired across physiologic conditions. And that also includes, obviously, analyzing how oocytes or egg cells persist in a non-dividing state, meaning they're quiescent. And that protects against damage during aging. So this work really has the potential, I would say, to help us understand how to preserve equality with age. Iain's also a spectacular mentor to his students and postdocs, and he is deeply committed to ensuring equity in science. So we've had some really fun conversations around that. So welcome, Iain. I'm excited to hear your talk.
Thank you very much. Yeah, And thanks for the fantastic introductions to both to this important topic today and to us as well. It's great to be here, and I'm really excited to be able to talk about what is a very important topic and also share with you some of the recent work. And I'm going to talk about the important cell that we've already heard about today, the oocyte, and particularly how we safeguard the fidelity of that oocyte during female reproductive aging, so that we can start things off well with a good chromosomal content of an oocyte. So I think often people end with the acknowledgements, and I wanted to actually start here with the acknowledgements today for several reasons, part because I think the people who do the work are really important, and also I love this group of people. I'm really glad that I get to work with this group of people every day. I'm going to, as Jennifer mentioned, our lab is generally interested in the process of cell division and the different physiological circumstances under which you have to think about the way that cells divide. The work I'm going to tell you about today was primarily done by a postdoc in the lab, Zach Schwartz. But for the work that we're doing moving forward, and particularly we're really proud to be part of this really fantastic consortium, the Global Consortium for Reproductive Longevity and Equality, that we're all here to recognize. And the work under that is not only being done by Zach, but also Ali Nguyen, Jimmy Lee, and Sarah Katie. And so together, this team is really interested in understanding this process that you heard about, which is how do you ensure the fidelity of chromosome segregation during meiosis when you form an oocyte? So the other thing I want to begin with is this picture here. So this is a karyotype. This is the chromosomal content of a single cell. And so what we're seeing here is a chromosome spread where you can see all 23 pairs of our chromosomes. And I think many of you have probably seen this image before. It's maybe not the best reproduction in the world, but it is one of my two favorite karyotypes in the world. And it is actually the karyotype of my older daughter. And so this is something that when she was still in her mom's belly, we had analyzed to see the chromosomal content. And it was actually the first time that we knew that she was a girl because there was two X chromosomes and not an X and a Y. But it's also something that we did because both of us were a little bit older and just thinking about making sure that everything was going to be great as she began to develop. And one of the reasons for this is this really pretty serious effect, which is the maternal age effect. I would say I'm not in love with these graphs and the way that we're presenting this information, but I want to highlight these two features that do happen during reproductive aging in women. And the first is that there is an increased incidence of some chromosomal abnormalities, such as extra copy of chromosome 21 that you would find in Down syndrome. And this definitely does increase over age. And so this suggests that at least a subset of oocytes that are made, when they go through the process of meiosis, the subsequent eggs that are generated may have chromosomal imbalances. The other thing that one sees during reproductive aging is that decrease in fertility coupled with an increase in spontaneous abortions. And actually, many of these come from a probably very similar source to this graph on the left. So there are only a few chromosomes where an extra copy of that chromosome is tolerated in human cells, chromosome 21 being one of those. Most of the times when you have some kind of missegregation effect during meiosis, you end up with reduced fertility or some kind of spontaneous abortion. And so we need to understand really the processes by which meiosis works and the way that this can go awry as we all age. So I want to highlight this point that Jennifer made really well here, which is that the oocyte is a remarkable cell in our body and that it's made so early in our development, but it's something that must persist for decades. And so it's made while you're still in utero. But if we take this oocyte out and here we look at it, it's initially formed. These cells are formed before a human is even born. And then that cell must sit there in this paused state, in a state called quiescence, where it's essentially hibernating, where it can sit for decades until it's triggered again by a hormonal stimulus. And when it does that, it receives a hormonal stimulus and it begins to mature, and then it will go through a process called meiosis that will be able to form a haploid egg that can be fertilized by a sperm. And so I'm going to show you what actually that process looks like when we begin again. So this cell is, again, sitting paused for decades. And that cell looks a lot like the one here. This doesn't happen to be a human oocyte, but the whole process, as I'm going to show you, look very similar. And in this oocyte is sitting here paused, and it's just about to be hormonally triggered in this case to begin the process of meiosis. And so we're seeing this round oocyte. The red is the DNA and the nucleus, and it's been labeled here by a red histone protein to mark the DNA. And then we're also going to be able to see the microtubule polymers, which are an important player in ensuring that these chromosomes can be captured and divide. So this process is going to begin. We're going to see hormonal stimulation here. And in this movie, the first thing that's going to happen is that the nucleus envelope will break down, and the chromosomes are going to essentially gather and capture on this spindle. And right there, actually, in this organism, we just saw sperm entry. And so the first process we see here is meiosis 1. We're going to segregate the chromosomes and actually get rid of half of them and exclude them from the oocyte into what's called a polar body. And then it just went through a second meiotic division very rapidly there. And then finally, this oocyte, the maternal pronucleus is going to emerge and meet the paternal pronucleus to form now, again, a diploid cell. And that cell itself is going to go through now what is a mitotic division to form the first two cells of what now is a new developing embryo. And so during this, you have to have these rapid division processes, those two very small spindle divisions to create, to get rid of most of the content and reduce you down from being a diploid to being a haploid that you can now be able to start all over again. And so if you imagine this process, coming back to my daughter's karyotype here, you start out with all 23 of these chromosomes, 2 copies of each. During meiosis, you have to be able to take it now so that you have a single copy of each one of these chromosomes. And those then are paired with 23 chromosomes from the father as well to make a new individual with 43 chromosomes. And so we have to think about the process of making sure that there are no imbalances. And that means that we can't leave a single one out. When we're undergoing this segregation process, we need to be able to capture each one of these chromosomes and ensure that it's resulting in the new nucleus that's formed in that new egg that begins there. And so we have to undergo this segregation process. And so we need a set of machinery that's going to enable that. So if we focus in on just one of these chromosomes, you can actually see here that there's sort of a pinch point, there's a constriction. And it is this region of the chromosome that is driving forward this process of its distribution to the two new cells. But this process actually is not the DNA that's driving it. It's in fact the protein machinery that's driving that. And so if we focus in on this with another image here, we can see these images of chromosomes in blue. There are sequences on each of these chromosomes. DNA sequences in us typically referred to as alpha satellite DNA. But really, it's the protein components that are marking these regions, marking the primary constriction. And it is these protein components that are actually mediating chromosome segregation. So for example, we have a specialized molecule. This is really the only name of a protein that I care about you remembering today. There's a specialized histone H3 variant, specialized nucleosome that's present at centromeres called CENP-A that's mediating this process. It's marking this site and it's directing things. So if we imagine what this looks like, instead of having to worry about the underlying DNA sequences, we just have to worry about where this CENP-A molecule is. This is the thing that's specifying where the centromere is. And so this protein here, CENP-A at centromeres, it can actually recruit a bunch of other stuff. And again, the names of these don't matter, but really you can build up this beautiful large segregation apparatus, this machinery that directs cell division and chromosome segregation. However, if we were to eliminate CENP-A, all of these other proteins would go away, and you would no longer be able to distribute and recognize a chromosome and segregate it. And even worse than that, once CENP-A is eliminated there, it can never come back to the site. Its process requires that there be pre-existing CENP-A. And so you have a really high premium on being able to maintain it under all costs. And I think this is actually one of the really big challenges that we face as we age. DNA repair can occur. We have a lot of knowledge about how you can fix issues with DNA, but proteins are really highly susceptible to age. They can receive oxidative damage and other things. And so we have to think about how we rejuvenate these structures in our cells. So there's been a dogma in this field for a while, which is that this you don't really have to worry about this problem because these CENP-A nucleosomes are just remarkably stable. And so if you look at them in a human cell and culture that's rapidly dividing, there's really no exchange. They're essentially infinitely stable. And so I think that all of the work in cultured cells is consistent with this. But what we've heard too is that these oocytes have to persist not just for a few cell division cycles, but really for decades. And so I think we face a very different challenge when we start to think about the process of maintaining these protein machinery in an oocyte over extended periods of time. So Zach wanted to be able to study this process, and in particular, he wanted to be able to monitor whether there's possibilities to replenish and replace this new CENP-A. And so the way that he did this is he took oocytes and he injected a fluorescent fusion to CENP-A to just be able to detect it. So the oocytes that we use for these experiments actually come from a marine organism. It's not a mammalian system, and I think many of the processes happening here are similar. And this is what we're focused on studying now with the funding that we're really lucky to receive from the GCRLE. So Zach not only looked at this, but actually he came up with a really nice strategy in which he could age these oocytes in vitro, and he could actually age them for what is not a long period of time for us, but is a long time in the lab, which is 14 days. And then he could watch this process and see, well, instead of this infinite stability that was assumed for CENP-A, how do they behave here? So when Zach saw that and he looked early on, it really is consistent with what the dogma in the cell culture field was, which is that there's just no ability to put in this new protein, and there really is no dynamics of this. But when he had the patients to wait, he could actually see that you could start to see these dots, start to see this fluorescent protein. And he actually could quantify this over time. And what he saw was just this really gradual increase in these CENP-A molecules. You're able to put down this new histone and retain the amount of CENP-A that's there over time. And this also has important consequences. So normally, if we age oocytes in culture, they're happy essentially kind of infinitely in this case, maintaining fidelity. But if Zach now blocks this new deposition process, the ability to replenish this centromere protein over time, now if he blocks that, you can see that there becomes really a major challenge. Instead of beautiful, nice segregation, now these chromosomes missegregate. And you can see a dramatic increase in the number of oocytes that display chromosome missegregation and problems. So if you imagine what's happening here, all of these chromosomes that are being missegregated right here are not going to make the way into the oocyte appropriately. Maybe there's not enough of a given chromosome or too many. And this is very consistent with what we see in terms of the chromosomal abnormalities I mentioned at the beginning, which is that small imbalances in missegregation can be quite consequential for the resulting egg and embryo. So to sort of summarize what I've showed you here, protein components in general are really susceptible to kind of damage that would happen during aging. And in particular, the processes that we've been able to explore here really suggest that instead of being infinitely stable, these nucleosomes can be kicked out sometimes. And this can be a problem. And if you don't replenish them, this can actually be a challenge. But the machinery that exists is able to come in and deposit new CENP-A. And this really distinguishes it from what we see in cell culture. And so there is actually an ability to rejuvenate the centromere. So if during aging you were to start to not be able to do the replacement process, that would be a serious problem for aging. But I think even itself, the ability to kick out one of these nucleosomes and replace with a fresh nucleosome is actually really important. And so you need a strategy to get rid of old and damaged proteins and replace them with new ones. And so a lot of our work too is focused on the mechanisms that promote this. And at least one of the hints that we have is that transcription plays a key role. And so although oocytes are normally thought to be pause cells that aren't really doing a lot, they're actually relatively active in their ability to drive ongoing transcription. These are just several different ways of looking at it. We can detect nascent RNAs and RNA polymerase there. And when Zach disrupts this and blocks the presence of transcription, then this really blocks new CENP-A turnover. And so we think this ongoing CENP-A transcription is really important for turning over centromeres. And we think this is true for this important cell division structure that we work on, but we actually think this is generally true. And so Zach actually sees the similar ability for all across the chromosome. All of the chromatin can be refreshed and turned over in an oocyte. But that process is blocked when you block transcription using an inhibitor, in this case, an RNA polymerase inhibitor, triptolide. So the last thing I want to mention is we really are interested in this process of rejuvenating the centromere during female reproductive aging. And I think for us, quiescent cells like the oocyte are quite important. They're not the only cells in our body that need to withstand long periods of time and that ability to divide again. And so actually the liver is another source of that. And so working in mice in this case, we can actually see that if you look at a liver cell, which if you resect it has the ability to divide again indefinitely, the centromeres are also rejuvenated and maintained there. In contrast, if we look at other cells in our body, such as muscle cells or heart cells, in this case, we know that there are restrictions for those cells for their division process. And in these cases, actually, the centromeres are not rejuvenated and they're lost. And so we really think that this ability to retain a centromere is a key feature in terms of ensuring the fidelity of a cell and its ability to divide. And so then lastly, again, I think both at the beginning and the end, thanking the people and the people who generously provide the funding is most important. I mentioned the work from Zach, and I'm really excited for the work that Sarah, Jimmy, and Ali and Zach are doing under this important funding. And it's really wonderful to be here as well. Thank you very much.
Thank you, Iain. This is a most interesting and fascinating talk, especially with all the interesting images on how the egg actually undergo this unique division in the cell itself. And egg is very special because it only carries half the number of chromosomes, so they can meet the sperm and give you the one full normal karyotype, as Iain has rightfully shared with us, and showing his daughter's karyotype, which is amazing. I remember I took almost a year during my PhD to understand and identify all the different chromosomes and how structured they are. And I think it's just a wonderful introduction because we get Bikem back online. And Bikem is a postdoctoral researcher in the laboratory of Dinah Layard at the University of California, San Francisco. And her previous work was on reproductive and developmental biology. And this has inspired her to broaden the scope to now better understand how female reproduction is shaped, particularly how duration of meiosis affects egg development, starting from early development through to the aging individual. So Bikem, I hope you can give us more information about how this unique division in the egg cell happens. And now to yourself. Present.
Thank you, Wei, for this kind of introduction. Let me share my screen. And as I was nicely introduced, that I'm a postdoctoral researcher in Dinah Layard's lab at UCSF. And today I'm going to talk about our efforts to connect early egg development to reproductive aging and why this connection is important for female reproductive health. Previous presenters in this webinar did a great job explaining how oocyte numbers dramatically decrease during development and aging. I would like to present this decline in a different way. So let's pretend that each dot in this slide represents an oocyte. So while fetal ovaries have millions of oocytes, there are about 300,000 oocytes in an ovary when a girl reaches to puberty. From this point, oocytes start dying off a rate of 1,000 per month, and of those, only one gets ovulated each month. A woman at around 30s often has around 100 and 150 oocytes in her reserve, and late into 30s, that number could be 25 to 10 per 1,000 or fewer. So you may think that 10,000 oocytes are still good enough when you consider that you need only one to conceive. However, while oocyte quantity goes downhill, the prevalence of chromosomal abnormalities and the incidence of miscarriage follow an opposite trend. It's fairly easier to explain the left side of schematic since this is more measurable and that's understood part. But when we talk about the competence, it's more challenging to understand how or why oocyte quality decreases with aging since we don't have many biomarkers to check. With aging, processes impaired may result into mitochondrial dysfunction, as well as a higher frequency of chromosome missegregation during meiosis. Of course, there are many potential factors that cause reduction of oocyte quality. Aneuploidy might be one and the leading one of them. Aneuploidy is having incorrect number of chromosomes in a given cell. For example, a cell having 45 or 47 chromosomes instead of the usual 46. Aneuploidies can lead to miscarriages or congenital birth defects, including Down syndrome, which in that case, individuals have an extra copy of one of their chromosomes. So what causes this? Basically, errors in chromosome segregation during meiosis can lead to aneuploidy. Of course, when we talk about aneuploid embryos, incorrect chromosome number can come from sperm or oocyte. And the aneuploidy rate in human sperm is about 1 to 8%, and this frequency doesn't change with paternal age. However, the aneuploidy rate in human oocyte increases from 20 to nearly 60% at later stages. So when you think about sperm and oocyte are actually originated from the same precursor cell, and they undergo the same cell division, which is meiosis, why do we see different error rates in the females? So chromosomal defects in oocytes can happen because of complicated feature of meiosis in the females. And I will try to explain this complicated feature, red and green lights. Germ cells in embryonic ovaries enter the meiotic initiation, like before the birth, as you see here in the green light. And meiosis progress for a while, and the first stop happens around the time of birth. With the increase in the hormone levels at the puberty, meiosis resumes and complete the first part of the meiosis just before the ovulation. And meiosis keeps progress until a certain point, and the second pause happens. And finally, meiosis gets completed only if an sperm fusion takes place. So in this slide, those dashed lines represent where meiosis pause. And although the second stop lasts around hours or days, the first stop persists in some oocytes up to 2 years in mice and 45 years or more in humans. And duration of meiosis in females provide a great chance for errors to happen and accumulate, which is actually the basis of the main model explaining the maternal age effect in the literature. So as I mentioned in my previous slide, germ cells enter meiosis before birth, as you see here, lovely embryonic mouse ovary, but not all germ cells enter at the same time. So germ cells located the anterior part of the ovary enter first, and it spreads to the posterior part. Production line model was proposed more than 50 years ago, and it basically says those oocytes enter meiosis first, as you see magenta or pink color. They get maturated first and ovulated earlier in the life when the individual is still young. But the germ cells enter meiosis last, that you see here in the light blue close to the posterior, follow the opposite route, and they develop later and get ovulated when the individual is still at advanced age. Based on this model, eggs produced earlier in the life tend to have better connections or crossovers or point of contact between the chromosomes than the eggs produced and ovulated later. Basically means that light blue oocytes have more faulty chromosomes, which can lead to miscarriages and developmental abnormalities. Although the production line is a long-standing model and there are supporting and opposing studies, in this model, all oocytes from the young ovaries and all oocytes from the old ovaries were isolated and they were compared, and this model was actually presented. This is not the best way considering the level of heterogeneity seen in various organs. Also, if the first entrants are located in the anterior part of the ovary, how further development of those oocytes, for example, follicle formation, happens in the middle part of the ovary? So all those questions in mind, in our previous study, we used a three-dimensional ovary imaging and analysis method, and we showed that the first meiotic entrants, those magenta cells, are actually in the middle of the ovary. While their distribution is biased to anterior, they are not necessarily at the anterior tip. With the algorithm we developed, we meticulously mapped them and we showed them actually there are more meiotic germ cells in the anterior compared to posterior, but we have some in the middle. And when we compare the meiotic germ cell numbers, edges of the ovary and the middle, again, we show there are more in the middle. And with the development progress, this meiotic wave actually expands to the periphery part, and by E14.5, all germ cells basically enter meiosis. And we asked another question, okay, we know there is a pattern in the meiotic initiation. What about the progression? Luckily, we had a marker that we can label those cells that you see in yellow color right now. And it actually, surprisingly, we saw that meiotic progression pattern doesn't follow the initiation. So that supports the idea that actually suggests that some germ cells spend more time in the earlier stages of meiosis. Considering the duration of meiosis in oocytes can lead to genomic instability, the question next actually becomes, does the timing of meiosis affect oocyte fate during development and aging? So we broke down this whole question into pieces and listed what we need to do actually to answer this big question. So we should label the first and last meiotic entrants in embryos. We should let them born and trace their development like through the postnatal development to the aging. And we should be able to actually compare the fitness and metabolism between two different oocyte populations to answer this complicated question. And we are trying to actually tackle an age-old question or model with new technological approaches that we are using a drug-inducible lineage tracing system to trace the first meiotic entrants in living mice. We generated this mouse line by using genetic engineering technologies. And when this mouse is crossed or mated with another specific mouse line which express red fluorescent protein in every cell, means every cell is going to be red. After the administration of the drug to the pregnant mom, all first meiotic germ cells should be turned to green. So then we can actually trace them during the development and aging. So this is our first result, that when we gave the drug really early during the development and we collected the samples at embryonic day E16.5, we are showing here that we are able to label those first meiotic entrants in green with the GFP expression, and you see the whole overview here and then the higher magnification. It is actually challenging to label precise population, but we are able to label them with remarkable precision, which around 6% of germ cells in the total populations are labeled with GFP. After we confirm that, yes, we can make those magenta cells into green, that we are actually analyzing first meiotic entrants' developmental timing, including their contribution to the oocyte pool, folliculogenesis, and recruitment for ovulation in the young adulthood and aging to determine their fates, where they are, and what actually they are doing. Actually, we can do the other thing, the vice versa thing, that by using another mouse line, that we can make those light blue last meiotic entrants green, and we can trace their development, including their contribution to the oocyte pool, folliculogenesis, and recruitment for ovulation in young adulthood and aging to determine their fates. So once we complete mapping of those two populations, first and last meiotic entrants, we are going to analyze whether meiotic errors between those two populations are different. So in the first meiotic entrance site, when we give the drug super early, that we can label them in the green, and we will let them develop until embryonic day E18.5, which is just before they're born in mice. And we can isolate those germ cells, and we will analyze the chromosomes to quantify the number of crossovers or defects. And with the last meiotic entrance sites, that we are labeling them, and we are again tracing until E18.5, and we will do the same analysis to quantify the number of chromosomes and synapsis defects. Last but not least, we are going to answer the question, does the rate of fertilization and developmental progression differ between those two populations? That we are going to use the oocytes developed from the first meiotic entrants versus the oocytes from the last meiotic entrants. And we will do in vitro fertilization which means that we will actually artificially do the fertilization, and we will analyze the fertilization rate and their embryo's further development progression to the blastocyst stage. And I think the coolest part with that, when we do all those identification or like the experiments, that we can ask the real question, are there any proteomic differences that actually can lead to identifying some factors that can be used in the clinics? So we are going to actually use oocytes from the first meiotic entrance versus the last meiotic entrance, and we will apply this technique called mass spec. Firstly, we will look at your general proteomic landscape, and then we will look for specific candidates like DNA metabolism, DNA repair processes, or antioxidant genes. And we will try to actually identify proteomic differences between two populations. And if there are any difference, that can lead to develop clinical biomarkers for use in assisted reproductive technologies in the future. And with that, I would like to thank this amazing group of people and generous funding. Specifically, I would like to thank Diana guiding me, actually letting me be an independent, but also being there when something goes sideways, which always happens. And the previous collaborators for helping me developing this 3D analysis technique. And specifically, Jennifer and Bevin from the Buck Institute, for their help in every aspect, and for creating incredibly supportive environment. And I think with that, I'm happy to hand over to hosts.
Thank you, Bikem. That was great. I really enjoyed the images, so beautiful, and the movies. Both of your presentations, Iain and Bikem, were really stunningly beautiful. So we do have some questions, and we want this to be a discussion. And so, if you both want to answer, that would be fantastic. Technically, this is a question for Iain. Does tetraploidy or polyploidy precede aneuploidy at any stage?
Yeah. And so, I'm looking forward to a fun discussion. Partly to say, too, the nice thing about these is we can see the participants, we can see who's asking the questions. And I'm excited about this question. I was also excited to see who's asking this. So this is from Karen Krasta, who was actually one of the people I'm a big fan of her and her work. And so Karen really is, been very influential in thinking about what the consequences of missegregation are and what happens to that DNA. And, basically the way in which a cell can become aneuploid, and tetraploidization for a cancer cell is a really key strategy. And so a cell fails in its division, it's going to fail to, double its genome copy number, and then that can really result in a lot of consequences downstream, and I think is a key source of things in cancer. I don't think that we have at least for our work, and my sense for the field in general, of issues that would arise during oocyte meiosis. I don't really have a sense that tetraploidization would be a problem. So if you fail to extrude a polar body, you could certainly end up with a tetraploid state where you had double the amount of DNA that should be there. I think that probably would result in failure of that oocyte to exist and mature. And so instead of a cancer cell being able to tolerate those dramatic chromosomal imbalances, I think that early on polar body exclusion failure can be an issue, but probably not in the development of aneuploidy.
Okay, Bikem, did you want to add anything?
That was correctly, completely, yes.
There are more questions from Karen. We can go through all of Karen's questions first. Thank you, Karen, for listening so carefully and putting in such great questions. So do you see micronuclei frequencies increased when CENP-A is lost?
This is the other thing that Karen has done really well. So basically, when you have a failure of segregation, our thought is mostly you put it in a nucleus. But basically what forms, a nuclear envelope breaks down as you go through mitosis or meiosis, and it has to reform when you finish that. And so you have to grab all those DNA together. And if you're missegregating DNA, if you have one mass of DNA in one place and one little one over on the side, it's actually going to form two different nuclei, a major one and then a micronucleus. And so the idea that micronuclei can form is also been a really key theme in the development of cancer. And I think actually this is one of those things that probably is understudied in the context of oocytes and meiosis and reproductive aging, because it's very hard to see those things. You have this massive cell, very large nucleus. And so if there was one small chromosome that was left on the side, I think it would be a much harder thing to see that. And to my knowledge, people have not looked at that closely. I think it's a really fascinating question and something that we as a field and actually Karen in her own work should check out too. But I would love to know what the answer to that is. And certainly I don't have that answer.
You have a beautiful visual behind you. So we can all imagine what it would look like if there was a bias in the size of the two different sides.
Yeah.
And there's one more question from Karen. Is the prophase arrest identical to that seen in a spindle checkpoint arrest? These are all really good questions, Karen.
And I like thinking about just those oocytes sitting for a while. And actually, from Bikem's work, I hadn't even really thought about this idea that the order in which you're making them could make a difference. And like that kind of blew my mind. I really enjoyed that too. The pause state is something where we tend to refer to this as quiescence. It is not going to be comparable to an arrest in mitosis, and it's also not going to be comparable to the other quiescence states that we would call G0 quiescence, the kind of places that the non-dividing somatic cells in our body exist. And so this prophase arrest does, other work from Zach, one of the things that we've done recently essentially map the landscape of phosphorylation across that paused oocyte and what happens when it changes, when it enters meiosis. And I think Bikem's work describing too, really, how one would use mass spec to be able to analyze that is quite important. That arrest state definitely does not look like what you would have for triggering the spindle assembly checkpoint. It also does not look like what you would see in G0 arrested cell. And so I think it probably is a little bit more like what you would imagine for quiescence, but unique to the oocyte in many other ways.
Yeah.
There's so many things that are unique, right? And this can last for up to 45 years, I think Bikem said. So I can't think of anything else like that in the rest of the body that could pause for 45 years.
Yeah, absolutely. And that brings to the nice point because Iain's and Bikem's work are actually very, very forward in that thinking, in trying to understand, why the ovaries and the oocytes are behaving in such a unique way. And that quiescence itself seems to accumulate many errors, right? As Iain has nicely bring out the protein part. And I think Bikem has now alluded that, the whole environment that the oocyte is developing in seems to be affecting how the oocytes stay alive for so long, although in a quiescent state. And there's this interesting question actually is asking, does migration across the ovary actually affect the ability to ovulate? And I think that's quite interesting because Bikem, you were talking about, those that get enter meiosis first, gets ovulated first. And that is interesting because how does the ovary knows that?
Oh, how does the ovary knows that? In terms of embryonic development, there is a unique spatial distribution 3D order as they enter development and then they progress. But I think the biggest problem with answering those questions, this is happening in the embryo. So to be able to understand those stuff that we need to use embryonic samples, which is not always super easy. And that's why there are so many questions. And I think the main question was environmental clues, or what was what was your first question?
How does this environmental cues and the mechanisms that affect the ovary to its ability to ovulate first? What comes first goes out first.
Ovulate, ovulation is such a complicated thing, but there are some extrinsic signals that they say, okay, go and develop. For example, enter meiosis. There are some extrinsic signals, but later ovulation is such a complicated process that under the hormonal control, and there are so many things that we don't know intrinsically how germ cells decide, okay, this is my turn to develop. So sorry, but there are not straightforward answer for those questions.
I see. So are you able to postulate any hormones that could be involved in this? Is it due to the gradient of hormones that you have mentioned? Any particular molecules that you think actually...
For ovulation, it can be actually not directly hormone, it can be vasculature, because actually vasculature kind of carries the hormonal cues to the ovary, or it can be maybe nerve innervation. But we really don't know right now, because we are lacking of this like location information when we analyze the ovary. It's almost impossible with humans, but like we are really trying to push the technologies to see, can we see the things in real time and then analyze them?
Right. Because I'm asking this question because it's like, is there a way to actually, as what Iain has so nicely put in as well, is there a way to rejuvenate the oocyte when it's in the quiescent state, whereby its source of nutrients, its microenvironment is always changing, but it's just held in that quiescent state,
Basically, when they are in primordial follicle stage, we know that there are some like environmental clues that they just stay in that stage and they don't altogether start development. But we just know there are some like stop signals to just preserve them not being developed. But we don't know what kind of clicks or lifts that signal, and then they start developing as a cohort of prenatal or later stages.
Right. Okay, that's all work in progress, and we need to find that out so I could give more answers to the ladies who are asking me this question every single day. and I think I want to move to Iain, because this is something quite particular, especially in terms of CENP-A, because rejuvenating it also depends on other factors such as CENP-C and T. How does these two proteins affect the functionality of CENP-A? This is quite technical, so
yeah, and maybe to make that large, so one of the challenges of working on cell division is that it actually is literally an alphabet soup. So we have CENP-A through Z basically, and another 70 proteins. And I think you give one of these talks, and it often you're focusing on the names and the numbers, and instead of thinking about the big pictures. And I think that this concept here that the proteins are uniquely susceptible to the challenges that they're faced during aging is quite important. And I mentioned just one molecule, which is this nucleosome, but it's not the only player that's there. And actually, that's kind of the grant that we wrote for the GCRLE is to be able to think about not only putting this much more in the context of mammalian oocyte aging, but also to think about sort of the other proteins and players and how they're changing during this process. And so this CENP-A molecule, which I like, and I think is a pretty good prototypical example of what's going on there. But that question really nicely highlighted that there's other molecular players that are also facing a challenge, and they're all really intertwined, and their functions are connected and interrelated in beautiful ways. And I don't think we have an answer for that. And so we've looked at some of those proteins, and we're starting to get a hint of how they might be changing both during this pause state and during quiescence. But is definitely forefront in our mind is that you've got to solve this problem for each of those factors, and to think about the bigger picture of how they're able to persist, withstand these challenges, and rejuvenate. And so I think that, a lot of the challenges of oocyte aging, so prior work, really beautiful work from Melina Xu and Mike Lampson and Kim Naismith and others, has thought about the challenges faced by the chromosomal glue. So the glue that essentially holds chromosomes together to enable meiosis is a molecule called cohesin. Also really important molecule that you need to be able to sort of protect and withstand. And if you had a way to rejuvenate that would be important. In our case, it's focused on these centromere proteins, and that question really nicely highlighted some where I want to know. And hopefully if you all do another series of these and you invite me back at some point, I'd hope to be able to tell you as well.
Definitely will want you back.
Yeah, for sure. So, okay, this is a thought question, and you can both answer it, but you're mentioning rejuvenation. So, can you draw some hypothesis about, how you think it's working, and then how do you envision actually, how would you rejuvenate the system or correct the defects if this is really truly where the causal errors are that lead to what we see later on? I know that's a hard question.
I will take the first step a little bit smaller. If we can identify them in the clinics from 45 years old patient that when clinicians collect the oocytes for this reproductive technologies, even in that point, if it's possible to identify them, saying that with really easy markers, oh, those are looking a little bit younger, those are looking a little bit older, even this can help a lot. With the rejuvenation, I can just think of the genetic manipulations, which is not possible right now. But I think this is the first thing that what easily can be done, and then next steps can be thought about, actually. And Iain, sorry, it was my first name, so.
Yeah, that's great. I think that one of the things that makes my head hurt about that question is that series of time scale. So, we in the lab, like, if the experiment takes more than 2 days, you're like, more than 2 days, right? And like, so for a lot of the quiescence experiments that we're doing in the context of the lab, it's been, say, 2 or 3 weeks, and which is a long time, to sort of be waiting for something to happen. But it's a pretty short period of time overall in the context of aging. When we think about aging, for us, the in vitro system that we had for oocytes is from marine organisms, as I mentioned, and we can see those changes happening over several weeks. In us, we're talking about decades, but when we do aging studies in mice, we're talking about, well, an aged animal is a year and a half or something like that. It's striking to think about what aging means when we imagine the oocytes are fine in this pause state for 2-3 decades. And what is changing at that point? And so in some ways, although they're susceptible to this aging process, and we really need to be thinking about that, the fact that they've done such a good job of maintaining that, fresh rejuvenated state for what is an unbelievably long time is pretty remarkable. And so I think that we need to capture the processes that must be happening there just in an ongoing way, and then think about how we can push and extend what that is to achieve what we would hope to be able to achieve, which is genuine that reproductive longevity. And yeah, it's a challenge. Like in terms of, I think the statement, I feel comfortable that cells can express things to repair DNA, but that you imagine the protein composition of an oocyte when it's made in a developing person. I suspect that most of the proteins that are present there will have been turned over by the time that oocyte actually acts. And so a long-lived protein is actually kind of a rare thing. And so there is really nice work from Martin Hetzer's lab at the Salk looking at long-lived proteins in the brain. There are not that many proteins that are that long-lived, and you can't turn over in that way. And so I think thinking about the strategies that a cell employs to be able to do that and to ensure that there is sort of that dynamic feature. And I think some cellular structure is going to be uniquely sensitive to that. And I think the centromere and cohesion are some of those, but there's definitely going to be others as well.
Yeah, I mean, well, this is why the consortium exists, right? That there's so much basic science that we don't know the answer to the questions. There was something that jumped out at me in Bikem's slides that I hadn't really appreciated before when she was drawing the distinction between the aneuploidy rates and how they increase with age in male versus female. And I didn't realize that the aneuploidy rate in young females starts at 20%. It went from 20 to 60%, whereas in males, it went from like 1 to 8%. So do you know what accounts for that huge aneuploidy rate, even in young females? 20% seems high to me.
Even in young females, oocytes start developing when those females were there in like their mother's womb. So even in females, actually, those oocytes have been sitting there for probably 20 years with like uncompleted chromosomal structure and like open for all the stress. And I think that is the major thing, that it's such a prolonged process, even if it's the same cell division. And even in the young females, we see that frequency. And I think I should add one thing with the developing technologies, that those percentages might change, but still oocyte aneuploidy is the greatest risk for aneuploidies in embryos.
Yeah, and I think that both your work are amazing because Iain is focused on the quiescent state of the cells, especially oocytes, is holding for such a long quiescent state of 20 years, and Bikem, you're looking at really the what is happening, who enters meiosis first, and what happens when they undergo the sex chromosome division first. But do you know during the process of what actually could have caused all the other oocytes to actually die at the same time? Remember, you both showed the slides that they all start to die. Do you think you can unravel that mechanism as well with the DNA, pathway that could the oocytes be doing? What is happening at that level?
This, I think, not only oocyte, but germ cell loss is a common feature of gametes in various species. In the males, that can happen a little bit earlier in the life. In the females, it happens a little bit later. I see this is such a huge waste of energy in terms of the organism, but we really don't know what is the main reason for this great germ cell loss. One cause might be actually those are the cells that actually pass your genetic information to the next generation, and maybe that's why they are in massive in numbers to just eliminate any error or just trying to promote I think the future generations, and that might be answered. But personally, I don't know why they are dying that many, and I don't think there's any certain answer for that.
Iain, any thoughts of that? Because
I can't possibly answer better than that. That was a great answer, and very, very thoughtful and really nicely consistent.
Because I was actually thinking from the translational point of view, in that case, could you just stop them from dying? So that would guarantee reproductive longevity, isn't it? We know
stopping them from dying may not be actually the good thing. Maybe this is the quality control. So we don't know. We don't want to have 10,000 more numbers, but in a really bad quality.
Absolutely.
So I think that's a kind of the thing. And also actually that brings a good point that during the female development, we know that some important organelles actually can be transferred from one certain oocyte to the another, which in the field generally is thought that it is a supporting mechanism for the future the oocyte. So but still it doesn't answer right away why they are dying in that high numbers.
Yeah. I mean, what you're saying, Bikem, I think, is that we just need to understand why some germ cells spend more time in the early stages of meiosis. Right?
Yes, easier steps first.
There'll be so much work to do.
Yeah, we're at the end of our time, but thank you so much, both of you, for wonderful talks and discussion. And I wanted to remind everyone that next week is a holiday in Singapore, and so we will not have episode 7 next week. We will have it the following week. And we're going to welcome, actually, related to what Bikem was just talking about, we're going to welcome 2 speakers, Lene Brayboy, who's going to talk to us about one of those organelles that seems to be very important for oocyte function, mitochondria, and Minhoo Kim, who's going to talk to us about the microbiome and female reproductive aging. So please tune in 2 weeks from now. And we'll continue this discussion. If you have more questions, if you'd like to chat with Iain and Bikem remotely, we'll continue this discussion on our network discussion page at gcrle.org.
Thank you, everybody. And I hope everyone will have a lovely upcoming weekend. And those in Singapore, have a lovely upcoming long weekend next week. We'll see you in 2 weeks' time. And until then, making reproductive longevity a reality. Thank you, everybody, for your attendance and your wonderful questions.